Hello, everyone. Welcome to Ripen the MeriSTEM Brain Awareness Week live question answering period. So it's also Women's Heritage Month, Women's History Month. So I wear this pin to sort of celebrate and bring attention to all the contributions that women have made to our country, as well as to science, technology, engineering, and mathematics. I am the founder and executive director for Ripen the MeriSTEM. And I also am a neuroscientist, and I also have chemistry training. And so today I wanted to bring some awareness about neuroscience, the brain, based on questions that have been posted on our various social media platforms. And so that's what we're going to do today. So if you're watching and, sorry, I'm sweating like a pig as I tend to do these days. If you're watching and you have questions about anything that I'm saying, feel free to post it here. Or if you're watching after the fact, you can feel free to post questions on the video, as well as email us at ripenthemeristem@gmail.com. You could also send us a DM. So I'm going to flip the camera around because I think it will help to have visuals for some of the stuff that we're talking about today based on the questions that have been posed. So I'm going to flip around the camera and I'm just going to get started on the questions that have been posed to me. All right. So as this shows, it's Brain Awareness Week 2026. And this is being brought to you by Ripen the MeriSTEM. It is a 501c3 that is based in Maryland. And we serve students and early career professionals who are interested in pursuing science, technology, engineering, and mathematics careers, or who are just interested in science, technology, engineering, and mathematics in general. So if you need any type of assistance in STEM, reach out to us. We'd love to help you. So the first question I received was, what types of stimuli can cause permanent brain damage? So before answering that question, I think it's important to give you a little bit of background that'll help you understand the basis for the answer to this question. So the first thing is that there are two classes of cell types in the nervous system. On the left, we have neurons, and there are many types of neurons in the brain and in the peripheral nervous system. And on the right, we have glia, and there are many types of glia. So neurons are the cells that send messages with neurotransmitters, and glia support neurons. So you have glia that act as immune cells. You have glia that help insulate neuron fibers to make signals spread faster. Glia serve a variety of functions. But what's important for today's question is that neurons do not replicate. So once they become neurons, if they die, they don't regenerate. So if a neuron is damaged or if it is killed, it does not return. On the other hand, glia are able to replicate. So this on the right is showing a cell dividing in two. So the problem with this is, obviously, if your neurons die, they never return. So you want to do everything you can to avoid neuronal death. Now, we're going to go through some of the common causes of neuron death. So we'll do many different slides related to this. So one of the first causes we'll discuss is disease. So you may have heard of Alzheimer's disease, which is a form of dementia. Not all dementia is Alzheimer's disease, but all Alzheimer's disease is dementia. It is something that affects our aging population, but also some people experience Alzheimer's disease in an early onset way. We have Parkinson's disease, which is a neurodegenerative movement disorder that also usually affects people in the elderly population. ALS, which some people refer to as Lou Gehrig's disease, which is a motor neuron degenerative disease. And then Huntington's disease is another movement-related disease. And so these diseases just represent a small portion of the many different neuronal diseases that can cause death. But all of these are considered neurodegenerative diseases, and they are slow and they progress over time. So I would say in terms of neuronal death, disease is one of the top at the list that cause death. The second thing is stroke. And I'm sure that you've heard that term. So basically, a stroke is an interruption in blood flow to the brain. And there are two types of stroke. At the top, you see the word ischemic, or you may have heard the term ischemic attack or ischemia. So that is the type of stroke that is caused by a blockage in a vessel. So you see, let's see if I'll grab the mouse. You see here, this cholesterol or fat or whatever this is supposed to be, that is impairing the ability for the blood to flow through the vessel that brings blood into the brain. So if this happens, then you'll get an area that is supplied by this vessel that will die because of a lack of blood flow to that region. Okay. The other type of stroke is hemorrhagic stroke, and that's when you have a blood vessel to burst. So if you have an aneurysm, which is when a vessel is expanded, sort of like balloons out abnormally, that can burst. And if it bursts, all of the blood that is within it will spill out into the brain. So one, that will disrupt blood flow to all the regions that that vessel supplies. But also, blood itself is not supposed to be in the brain. And it's toxic when it comes in contact with the neuronal cells and glial cells. So those are two types of strokes, or the two major types of strokes. And yes, stroke causes irreparable damage. Although sometimes your brain can compensate for it over time, but initially you're going to have death of neurons because of stroke. Another common cause of neuronal death is pathogens. So viruses, bacteria, even fungi can cause neuronal death. So these are just some common diseases that are caused by pathogens. Meningitis, which can be viral or bacterial. There are HIV-related neuronal deficits and illnesses that are associated with the death of neurons. Malaria can cause death. West Nile virus, syphilis virus, herpes simplex virus type 1 can also cause death. And this is just a short list of some of the diseases I thought you may recognize that can cause death of neurons. The next thing we'll talk about is injury. You may have heard of traumatic brain injury, which is head trauma, and it could be caused by a variety of things. You could be a boxer and undergo lots of blows to your head. You could get in a car accident. You could have a motorcycle accident. There are numerous things that can cause trauma to the head, and that trauma can cause death of neurons in regions that are affected. You also can, obviously, if you suffer a gunshot wound to the head, any neurons that are in the way of the bullet will die and will never return. Concussions, which is head trauma, essentially, can cause death in some cases. And then a specific outcome, if you will, of head trauma is chronic traumatic encephalopathy, which is known as CTE, and is something that we've discovered in football players years after they're no longer playing football. So injury to your brain can cause death of neurons. And yeah. Another cause of death, and this is something that may be of interest to a lot of people is chemical exposure. And so there are a variety of chemicals. So when I say chemical, I'm talking about alcohol use, illicit drug use, specifically methamphetamines, MDMA, which is ecstasy, cocaine drugs are known to cause death of neurons. Then there are certain heavy metals like lead, mercury, manganese, arsenic, and cadmium that are known to be associated with the death of neurons. Certain pesticides and herbicides that you may be exposed to on food. I've done some research in the lab with some pesticides, and we know that some pesticides are capable of killing neurons directly and effectively. And then, I hate to break it to those of us who are smokers, but tobacco smoke, exposure to tobacco smoke, whether you're smoking a cigarette or you're getting secondary exposure to someone else smoking, tobacco smoke can kill neurons. And then we have miscellaneous causes of neuronal death. So stress, sleep deprivation, hypoxia, which is low oxygen, the lack of oxygen. So people who have sleep apnea, they would fall in the category where they're not breathing at different points during the night, and the reduced oxygen traveling to the brain causes death of neurons over time. Extreme heat can kill neurons. Extreme dehydration can kill neurons. Malnutrition, so there are certain vitamins that are needed for your brain to be healthy, your body in general to be healthy. So if you're not getting all the proper nutrients, proteins, the sugars, then your brain can suffer and neurons can die. And hormone disruption. Also, there are certain hormones needed for your brain to function. And, if your hormones get imbalanced or these hormones get imbalanced, it can kill neurons. And there's some other things as well. But this is just sort of things that I figured most people would recognize. All right. So the second question I got, and I only got 2 questions that I wanted to put into the presentation. I got another question that is a more personal matter, so I didn't feel the need to put it here, but I did answer that question for that person. But the second question was, what can we do to optimize brain function? So again, in order to make it clear, I wanted to do a little bit of background to help you understand the way the brain works and the way neurons work. So first thing I want you to look at is here on the left is a depiction of a brain. And then you see these little lines. These are supposed to be parts of neurons that are coming out of the brain. But if you look at it, it kind of looks like circuits. So here I'm showing a circuit and how they look similar. So we in the neuroscience field refer to the connections of neurons as circuitry because they're basically like wires that send signals and that are connected to other neurons that receive the signal. And then the signal propagates throughout the body or throughout the brain by one neuron communicating with another neuron and that neuron communicating with the next neuron. So it is like circuitry. So over here to the right, I'm showing 3 different neurons. They're very similar types of neurons. But just to give you a sense so the way neurons work, they are what we call polarized, which means one side of the cell looks very different from the other side of the cell. And so if you look at this one, this part of the cell is the cell body. So if you took biology, you learn about the cell. So you'll have a nucleus, which is what this is supposed to be. You'll have mitochondria, and endoplasmic reticulum, and all the different organelles that most cells have. And then a neuron has these little processes that you see sticking out here that look like hair. These processes, when they're attached to the cell body, are called dendrites. And dendrites are the part of the neuron that receive the signal from other neurons or from other cells. And then if you look here, you see this long thing and then these little squiggly things at the end of that. So this part of the neuron is called the axon, right? And axons are actually coded in something called myelin, which is supplied by a glial cell type. So that's what this red thing is supposed to be. So the glial cells wrap around many times around the axon, and that allows a signal to propagate without leaking out. And then the signal reaches to the end, and the end of the neuron, this is the end of the axon, will release its signals from here, and then it can be received by the next neuron. So here is the cell body and the dendrites of this neuron, the axon, the end of this axon. So this axon can go on to connect to something off the screen. And so you can see these two neurons connect to this one neuron. So if you would imagine like looking up this circuitry, I don't really see in this particular circuit where two wires are connected to the one same wire, but imagine the axon is kind of like wires that are sending electrical signals. And in the case of cells, the electrical signal, it's not electrons, but it's actually sodium ions and potassium ions that are flowing through in and out of the neuron. And so the change in voltage, we call it, or the change in the flow of these ions in and out of the cell, the energy from that is actually used to cause the release of chemical messengers that are stored in, and I'll show you a picture of what that looks like, down here at the end of the axon. And then once those messengers receive the signal from up here, then they release the chemicals out in the space outside of the neuron. And then that chemical that's released can then bind and interact with this next neuron. So that's the way neuron communication occurs. And, I'm not really using jargon, so I could have said action potential or talked about the soma and axon hillock and a lot of different things. But in general, it's just important for you to know that neurons have two different sides. One side receives signals, the other side sends signals, and that they communicate with chemical messengers, but they also propagate like electrical signals that are composed of sodium and potassium. Okay, so in continuation, I'm showing here a blown up magnified version of, so on the previous slide, I showed, one neuron connecting to another neuron. So imagine if we were to amplify or magnify this region here, and then this is what we would see. So this is the axon region, let's imagine, from this neuron. And then this is one dendrite from this neuron. And so what happens is I talked about chemicals being released from the axon. So the chemical I'm referring to is called a neurotransmitter. So you may have heard of neurotransmitters, things like serotonin, dopamine, norepinephrine, glutamate. These are types of neurotransmitters. And in a neuron, neurons usually only have one type of neurotransmitter that they produce. So let's say this is a dopamine neuron. In a dopamine neuron, and in all neurons, in a dopamine, let's say a dopamine neuron, the dopamine would be represented by these little pink dots. And they are stored while the cell is at rest, if you will, in these little packages that are called vesicles. So in response to receiving a signal from up here, when the sodium is traveling in and the potassium is traveling out, and the sodium, that propagates all the way down. And then when it reaches the end, so let's say here's the end, the change in the voltage that happens, or the change in the concentration of the sodium ions that happen, there are proteins that detect that change in voltage. And when those proteins are activated, it allows for these vesicles with the dopamine neurotransmitter in it to say, hey, it's time for us to release our dopamine. So then the vesicles, which are made out of the same material as the plasma membrane of the axon, those vesicles move and bind to the surface of the axon, and then the contents are released out into this space here. So this space between the axon and the dendrite of two different neurons is called the synapse. Synapse. And so in neuroscience terms, we call this the presynaptic neuron and this the postsynaptic neuron. So because the synapse is here, this one is before the synapse, this one is after the synapse. Once the neurotransmitter is released into the synapse, so you see these little pink dots, then it has the opportunity to bind to these little pink things here. These are supposed to be receptors. Receptors are a special class of protein that in some cases, depending on the neuron, the receptor will bind the neurotransmitter and the receptor itself will change conformation and allow for the entry of ions into this cell. And then depending on what that ion is, it causes changes in this cell that can cause the propagation of another electrical signal in this cell and so forth, right? So that can happen. But there are also different types of neurotransmitters or different types of ions that can inhibit this secondary cell and turn off whatever signal is happening in this cell. So then downstream of that, the signal will be turned off for the next cell. So the propagation of this signal, the propagation of the signal here, causing the release of this neuron here, if it just happens one time, okay, that's what's supposed to happen. Normally, for a period of very short period of time, milliseconds, the neurotransmitter is in the synapse, it binds, and then there are glia, which aren't depicted here, that will clear the synapse of excess neurotransmitter so that this secondary neuron, this postsynaptic neuron, can be ready to receive another signal. If you allow for the neurotransmitter to continue to just be out in the synapse, eventually you'll get what we call desensitization of this neuron because all of its receptors will be bound to neurotransmitter. And then if more is released, then you will not have the opportunity to regulate this secondary or this postsynaptic neuron. So the objective is release the neurotransmitter, have it bind to the secondary or the postsynaptic neuron, clear the cell of the excess or clear the synapse of the excess neurotransmitter. And then the neurotransmitter that's bound here will do whatever it's supposed to do, and then it will be cleared. Okay. Well, the more this happens, let's say this neuron is stimulated many times, and it's always neurotransmitter in this synapse, and this neuron is continuously receiving this signal. Sometimes that can cause something we call long-term potentiation, or basically changes in the number of receptors that are present in the postsynaptic neuron. So, long-term neuronal changes or long-term neuronal synaptic changes, we refer to in neuroscience as neuroplasticity. And so, as I said before, if there's constant stimulation from this presynaptic neuron, which causes stimulation of this postsynaptic neuron, sometimes the postsynaptic neuron will make changes to accommodate all of the stimulation from the presynaptic neuron. And sometimes that change causes the increase of receptors in the surface of the postsynaptic neuron. So that means now I can accommodate more neurotransmitter. And this change is long-term. And long-term could be hours, it could be months, it could be years. So the more you stimulate this neuron, the more this neuron is stimulated, then there is a change in the synapse, and we call it neuroplasticity. Okay? So neuroplasticity occurs throughout your life. So the more you use something, the more change can happen. Okay? So we call this, we have a little term that neurons that fire together, wire together. So if you have neurons that are constantly being stimulated in the same pathway, then those neurons also will undergo changes that will affect the circuit that those neurons are in, right? So neurons that fire together, wire together. Another way to put this is use it or lose it, right? Practice makes perfect. So the more you practice something, let's say if you want to be good at playing the piano, you know, when you first start out, your brain is not exposed to this. So it may be difficult for you to learn to read the music, learn where to place your fingers. But when you practice it continually, you know, each day for a short period of time, or if you want to do for long periods of time, you will develop memory. And some people call it muscle memory. But yes, there is muscle memory. And then you also will have recall memory. And those memories will be strengthened. And over time, that's what we call learning. So when you use your brain or you use a certain circuit, that circuit is going to be strong. And then if you fail to use a circuit, that circuit will weaken. And if you allow a long enough period of time to pass, maybe that circuit will shrink and go away altogether. Meaning that the way that the neurons are wired changes because you're not utilizing that circuit. Your body and nature, we want to use energy wisely. So we're going to dedicate energy to what it's needed for. And then if we don't need it, we're not going to use it. We'll take that energy and apply it to something else. So use it or lose it. So if you want to know ways to help your brain, to optimize your brain to do different activities that you maybe want to be good at, or let's say you want to perform really well on a test, or there's something that you need to do and you need to be at your optimized level. It's very simple. Unfortunately, I don't have any like magical advice to give you, but your brain requires certain things: sleep, water, minimal stress, regular exercise, which increases circulation. So that's supplying blood to the brain and nutrients to the brain. And you want to avoid heavy meals. There are certain things that you eat that can cause you to be sluggish, and then it'll take energy away from your brain. So these are the ways that you can help your brain to optimize your brain. And if you have any questions about anything I said, if you want me to go into more detail about anything, feel free to contact us at Ripen the MeriSTEM .odoo.com. That's our website. Or you can email us at ripenthemeristem@gmail.com. And of course, you can follow us here on Facebook. Like this video, share this video. We also will be uploading it to YouTube. We have a YouTube channel, Ripen the MeriSTEM. We have a LinkedIn page. And yeah, hopefully this has been helpful. And so, like I said, yes, I'm a neuroscientist. I can answer questions in more detail. I can use more jargon if you'd like, but just let me know. I'd be glad to answer any questions you might have, or if you're interested in learning how to become a neuroscientist, or if you're wanting to get some internship or research experience related to the brain, reach out to me in honor of Brain Awareness Week. We can turn this into Brain Awareness Year and Brain Awareness Life. So hopefully you enjoyed this, and I wish you well. Signing off.